Energy considerations. Introduction to metabolism. What is metabolism? Metabolism refers to all chemical reactions occurring within an organism to sustain life. So the organisms which are occurring within an organism to sustain life, all these reactions are known as metabolisms, and these reactions allow cells to grow, reproduce, maintain structures, and respond to environmental changes. Metabolism involves energy transformation in living organisms. Number one, energy in biological system. Living organisms follow the laws of thermodynamics. So we got two laws of thermodynamics. The first one is the energy conservation. The second one is the entropy increase, second law. So in the first one, we say that energy cannot be created or destroyed, but it can be only transferred from one form to another form. For example, let's suppose chemical energy and glucose is converted into ATP. And second law, entropy increase, energy transformation increase disorder. For example, heart release during metabolism increase entropy. Types of metabolism reaction. We got exergonic reaction, and we got endergonic reaction. Exergonic reaction mean energy releasing, while inorganic reaction mean energy consuming. So releases energy, stores in chemical bond. For example, cellular respiration, glucose breakdown into ATP. While inorganic reaction, it gets energy, requires energy to build complex molecules. For example, photosynthesis uses sunlight to synthesize glucose. This is like the light energy coming from the sun in the chloroplast, and plus carbon dioxide plus water again through the cellular respiration, and all these combines in the chloroplast, and it gives us oxygen and glucose, which is then used in the mitochondria to generate ATP. Types of metabolic pathways. We got catabolic pathways, we got anabolic pathways. Catabolic and anabolic, what is the difference between them? So catabolic or energy releasing reactions break down complex molecules into simpler ones, release energy stored in chemical bonds. For example, cellular respiration breaks down glucose to produce ATP. While anabolic pathways, energy storing reactions. For example, it builds complex molecule from simpler one. So catabolic, it breaks down the complex into simpler one, while anabolic, making it the opposite. Now energy carrier in metabolism. So we got three energy carriers: ATP, which is direct energy source for cellular work, NADH and FADH2, electron carriers in respiration, and NADH, pH, which is electron donor in photosynthesis. Energy and metabolism. Metabolism is the sum of all chemical reactions that occurs in a cell to maintain life. These reactions require energy to build complex molecules or release energy by breaking down molecules. Catabolism. Now, energy in biological system. What is energy? First of all, what is energy? So energy is the capacity to do work. Capacity of the body to do work is known as energy. In biological system, energy is required for cell growth and repair, transport of molecules across membrane, muscle contraction movement, and enzyme activity in metabolism. Now, types of energy in cells. The first one is kinetic energy, energy of movement. For example, muscle contraction, molecular transport, potential energy, stored energy, chemical energy in ATP, and then chemical energy, energy stored in molecular bonds, for example, glucose ATP. Now, thermodynamics in metabolism. We got two laws of thermodynamics. The first one is energy conservation, and the second one is entropy increase. So conservation law, first law of thermodynamics says that energy cannot be created or destroyed, but it can be only converted from one form to another form, while the second law says that energy transformation increases disorder entropy. For example, some energy is also always lost as heat during metabolic reactions. Gibbs free energy. It determines whether a reaction requires energy or release energy. So formula is change in G is equal to change in H minus T delta S. The change in G if change in G is less than 0, that means that it is exergonic reaction, energy releasing spontaneous. If change in G is more than 0, that means that it is inorganic reaction and it requires energy. Now, glycolysis. What is glycolysis? Glycolysis is the first stage of cellular respiration. Where one glucose molecule is broken down into two molecules of pyruvate. Remember, we got one glucose molecule and it is converted into 2 molecules of pyruvate. So this is the definition. It occurs in the cytoplasm of the cell and does not require oxygen, making it a common pathway for both aerobic and anaerobic respiration. So it does not need any oxygen. The phases of glycolysis. Glycolysis consists of 10 enzyme catalyzed reaction divided into 2 main phases. 10 enzymes are working together to do in the glycolysis. Number 1 is energy investment phase, and the number 2 is energy payoff phase. So in the first one, the body actually invests the ATP, the energy, and in the second one, the body gets the energy. So glucose is phosphorylated twice using 2 ATP molecules. This forms fructose 1,6 biphosphate, and which is then split into 2 3-carbon molecules like G3P, glyceraldehyde 3-phosphate. You will see this on the next page. And then energy payoff phase. Energy payoff phase, G3 molecule undergoes oxidation, producing NADH and ATP. A total of 4 ATP molecules are synthesized by substrate level phosphorylation in result. So here we use 2 ATP and here we got 4 ATP. So the net result would be 2 pyruvate molecules plus 2 energy and a net gain of 2 ATP. So we got 2 ATP in the process of glycolysis because we got 4 but we also used 2. So 4 minus 2 is equal to 2. So the net gain is 2 ATP. These are the steps of the glycolysis. So glucose is first converted into glucose 6-phosphate, then fructose 6-phosphate, and in every step there is like enzyme involving. Then it is converted into fructose 1,6 biphosphate, then DHAP and G3P, and then it is it gives NAD plus NADH, and then in the 7th step again 1,3 base phosphoglycerate and 1,3 base phosphoglycerate is converted into 3-phosphoglycerate and then 2-phosphoglycerate, and then it is again converted into, and then it is converted into pyruvate. So you see here 1 ATP is used here, 1 ATP is used here, and we got 2 ATP here and 2 ATP here. So overall we got 2 ATP in that gain. Fate of pyruvate. Now what is the fate of pyruvate? Aerobic condition, oxygen present. Pyruvate enters the Krebs cycle in the mitochondria, leading to ATP production. So pyruvate can make ATP in aerobic respiration. Anaerobic respiration, no oxygen. Pyruvate undergoes fermentation. In the anaerobic respiration, in animals, the alcohol fermentation is yeast. It undergoes fermentation like lactic acid fermentation in animals, or alcohol fermentation in yeast. So net energy yield of glycolysis: 2 ATP are used, 4 ATPs are produced. NADH 0 are used and 2 are produced. So 2 NADH net gain and 0 pyruvates were used and 2 pyruvates are produced. So net gain is 2. The transition reaction, pyruvate oxidation. Now what is pyruvate oxidation? Overview. It occurs in the mitochondrial matrix. It converts pyruvate from glycolysis into acetyl coenzyme A. So the pyruvate which we got as an end product in the glycolysis, now the transition reaction, it will convert the pyruvate into acetyl coenzyme A. Steps: pyruvate transport, and the number 2 is decarboxylation. Number 3 is NADH formation. Number 4 is acetyl coenzyme A formation. Alright, so pyruvate enters the mitochondria from the cytoplasm. Decarboxylation, 1 carbon is removed from pyruvate, releasing carbon dioxide, and then NADH formation. The remaining 2 carbon fragment is oxidized, transferring electron to NAD plus equal to NADH. And acetyl coenzyme A formation, the 2 carbon molecules bind with coenzyme A to form acetyl coenzyme A and products per glucose. So we got 2 pyruvate and 2 acetyl coenzyme A enters Krebs cycle, 2 NADH goes to electron transportation, and 2 carbon dioxide releases waste. So the products of 2 pyruvate are all these 3. Citric acid cycle, Krebs cycle. Now this is another pathway. It occurs in the mitochondrial matrix. Acetyl coenzyme A enters the cycle. Acetyl coenzyme A enters the cycle, undergoing a series of enzyme catalyzed reaction. It produces NADH, FADH, ATP, and carbon dioxide. So in citric cycle, we get NADH, FADH2, ATP, and carbon dioxide. Now steps of the Krebs cycles. Steps. Now we will study steps of the Krebs cycle. Alright, so acetyl coenzyme A combines with oxaloacetate 4C to form citrate, six carbon compound. Then because 2 carbon are here and 4 carbon are here, so it will give us 6 carbon compound. Then citrate undergoes rearrangement and oxidation, releasing carbon dioxide. Energy carriers NADH and FADH2 are generated by electron transfer. Now ATP or GTP is synthesized through substrate level phosphorylation, and in the last step oxaloacetate is regenerated, allowing the cycle to continue. Now products per glucose cycle. So 6 NADH, 2 FADH2, 2 ATPs, and 4 carbon dioxide. All of them are released. This is the Krebs cycle. It first starts with the oxaloacetate. The citrate is converted into isocitrate with the equinetase enzyme, and then isocitrate, the carbon dioxide is released and it is converted into alpha glucotarvate, and then succinyl coenzyme A, then succinate, and then fumarate, malate, and oxaloacetate. You can see this diagram, but here in every step you can see here the NAD is converted into NADH, here the NAD is converted into NADH, the GDP is converted into GTP, FAD is converted into FADH2, and here NAD is converted into NADH. So this is the overall reaction of the Krebs cycle. Electron transport chain and accelerator phosphorylation. It occurs in the inner mitochondrial membrane and uses NADH and FADH2 to pump H+, creating a proton gradient, and ATP is produced with chemiosmosis using ATP synthesis. Remember, chemiosmosis is the main type where ATP is produced here. Oxygen is the final electron acceptor, forming water. Steps of electron transport chain and chemiosmosis. So electron is transferred, NADH and FADH2 donates electron to produce to protein complexes. Proton pumping. Electrons travel through electron transport chain, pumping H+ ions into the inter membrane space, and then proton gradient formation. A high concentration of H+ ions build up, creating potential energy, and ATP synthesis. Chemiosmosis. H+ ions flow back through ATP synthesis, driving ATP production. And the last step is oxygen is final electron acceptor. Oxygen combines with electrons in H+ to form water. The products are for 34 ATP. Majority of ATP comes from this stage. Remember, majority of the ATP comes from the electron transport chain in our body. 34 ATP is produced, and 1 water molecule is a byproduct. This is the summary. In glycolysis, we get 2 ATPs and 2 NADH and 2 pyruvate. In transition reaction, 2 NAD, 2 acetyl coenzyme, and Krebs cycle, 2 ATP in 6 NADH, 2 FADH2. And in electron transport chain, we get 34 ATP and water and ATP. So overall, we get 38 ATP energy for the cell, which is the energy for the cell. Fermentation. What is fermentation now? Fermentation is basically an anaerobic without oxygen process. So it happens without oxygen. So this process is without oxygen, in which cells generate energy when oxygen is scarce or unavailable. When oxygen is not available, usually it happens during exercise as well, when there is less, like when the cell cannot make enough amount of energy, so then we use fermentation. It occurs in microorganisms, muscle cells, and some plants, providing ATP through the partial breakdown of glucose. Overview of fermentation. It occurs in the cytoplasm of cells. It produces ATP through substrate level phosphorylation, much less than aerobic respiration, and recycles NADH to NAD+, allowing glycolysis to continue. Now, there are 2 types of fermentation: lactic acid fermentation and alcoholic fermentation. So if it is in muscle cell or bacteria, then our cells do lactic acid fermentation, and if it is yeast and bacteria, then it does alcoholic fermentations. Both the process are fermentation, but in different organisms it happens indifferently. Process of fermentation. So fermentation begins after glycolysis, which produces 2 ATP molecules and 2 NADH molecules. Lactic acid fermentation. Now the first one is lactic acid fermentation. So its equation is glucose is first converted into C3H6O3 plus 2 ATP and then it is so the end product for us is glucose is converting into lactic acid plus ATP. As you can see here, the glucose is converted into lactic acid plus ATP, and then steps. Glycolysis produces pyruvate, ATP, and NADH. Pyruvate is converted into lactic acid. Remember, saying glycolysis, if you remember, we got pyruvate, ATP, and NADH, and here the pyruvate is converted into lactic acid, regenerating NAD plus for glycolysis. It occurs in the muscle cell during intense exercise, causes muscle fatigue. For example, human muscle cell during oxygen debt, and bacteria in yogurt production. Lactobacillus. Alcoholic fermentation. Its equation is the glucose is converted into C2H5OH to alcohol plus carbon dioxide plus ATP. So glucose is equal to ethanol plus carbon dioxide plus ATP. And steps: glycolysis produces pyruvate, ATP, and NADH. Pyruvate is converted into ethanol and carbon dioxide, regenerating NAD+. It occurs in yeast and some bacteria. So it occurs in yeast and in some bacteria, and examples are yeast and baking bread making, CO2 makes the dough rise, and ethanol producing in alcoholic beverages. So let's talk about fermentation versus aerobic respiration. So oxygen required in fermentation, no oxygen, and aerobic, yes. Location in the cell, cytoplasm. For fermentation and for aerobic, it's cytoplasm plus mitochondria, and ATP yield 2 ATP here, and in aerobic 36 to 38 in product are lactic acid or ethanol, and here carbon dioxide plus water. And the efficiency of fermentation is very low because it gives you very less amount of energy, while the efficiency of aerobic respiration is very high. Importance of fermentation, we got some industrial application and we got some biological importance. So food production, yogurt, cheese, bread, wine, beer, and biofuel production, for example, ethanol from fermentation. And biological importance, survival of anaerobic organisms. It allows ATP production during oxygen shortage.